Hey guys, Dr. Israel here, and we're going to be doing a review of the first presentation in exercise physiology. This will be about a 15-minute lecture, just getting the basics, the most fundamental concepts. I'll be breezing through the slides, get you guys a good review in so you can be ready to take your quizzes and tests and do really well in the course. Alright, here we go. So we've got pictures of muscle people, excellent. We know this is going to be about muscles to some extent. So the general structure of a muscle fiber is very important to understand. First of all, most muscles insert into a bone via a tendon, and the outside of the tendon connects to what's called the epimysium, or the external casing of the muscle fiber. Inside the epimysiums are multiple muscle fascicles that are coated with an endomysium, so one level in. And of course, all the way inside, each individual muscle fiber or cell is also existing in each fascicle. So, just a quick summary. I screwed up already. The paramysium is what encodes the fascicle. The endomysium is what actually encodes the muscle cell itself. Alright? So the average muscle cell is actually very small, multiple composed muscle fibers. Some of the longest ones are 4.7 inches in length, usually occurring in the sartorius muscle of the leg. Alright. So we have some more diagrams here, some important terms for you. So the sartolemma is what's usually called the plasmalemma in a normal cell. It's the membrane that surrounds the muscle fiber or the muscle cell itself. The myofibril is essentially the component of a muscle cell, and a muscle cell is composed of multiple myofibrils. The sarcoplasm is the fluid inside the muscle cell in which everything goes on. So the sartolemma encloses the sarcoplasm. Inside the sarcoplasm are multiple myofibrils. Right? And of course, throughout this whole structure, this whole cell, we have T-tubules, and the T-tubules are actually tubes that take the extracellular environment and thread it all the way through the muscle cell. The T-tubules come in really, really important later when we look at how muscle contraction is initiated, because it's through T-tubules that we can send signals deep into the muscle cell. Some muscle cells are very, very large, and if we just communicated through the surface of the muscle cell, we wouldn't get communications deep into the cell. So T-tubules are essentially invaginations, or tubes that go all the way inside, deep into the muscle cell itself. Right? And the sarcoplasmic reticulum is in an animal cell known as the endoplasmic reticulum. It's primarily in the muscle cell designed to store calcium, and it releases calcium at the right time for muscle contraction to occur, and we'll look at that process in just a bit. Alright, so here you go. We have those T-tubules visible, and of course the individual myofibrils inside the sarcoplasm, encoded inside the sartolemma itself. Right? And that's your typical muscle cell, and you guys have these presentations so you can take a look through them and study this in greater detail. So, each muscle cell is composed of multiple myofibrils, and each myofibril is like a cable. And each section of the cable, one after another, is called a sarcomere. Alright? Sarcomere is the basic functional unit of the muscle fiber. That is, it is the smallest thing in a muscle fiber, or a muscle cell, that actually performs contraction. It starts with what's called a Z-disc, which is a structure of connective proteins, and goes all the way to the next Z-disc. And inside, there are a bunch of I bands, A bands, H zones. We're not too worried about that at this level of detail. But in order to hold these Z-discs together, in order to hold these sarcomeres, we have two very important proteins, titin and nebulin. These proteins are connective proteins. They don't actually cause contraction, but they hold the entire cell together, so when that contraction occurs, the rest of the cell gets pulled, and not just the sarcomeres by themselves. So there we go, Z-disc to Z-disc, and all of these sarcomeres add up to form myofibrils. You jam enough myofibrils next to each other, and you encode them with a sartolemma, and you get a muscle cell itself. So, of course, we have all that detail here. Now, really important stuff. Essentially, the two most important proteins in a muscle cell, not an over-exaggeration, are actin and myosin. Actin is also known as a thin filament, myosin as a thick filament. So, what do? Well, actin and myosin, here's actin, and it's coated by troponin it's coated by tropomyosin with essentially troponin holding that tropomyosin in place at various places. So actin is essentially a rod-like protein, it's a cable-like protein, and it interacts with myosin. Question is how? Well, exactly like this. Each one of these on each side is a Z disc. So the actin here is actually positioned in contact with the Z disc, it's holding the Z disc, so that wherever that actin moves, that Z disc will move. It pulls the Z disc along. Now myosin sits in the middle, in between each actin, all right? It's actually a 3D configuration. So myosin is in the center and it's surrounded by actins. All those actins are connected to the Z disc. What myosin does is each myosin molecule pulls actin, essentially like tug of war, into itself. So this actin will go that way, that actin will go that way, and what you get is contraction of the Z discs. Because titin and nebulin anchor the Z disc to the rest of the muscle cell, once the Z discs slide together by myosin pulling on actin, we get a contraction of the entire muscle fiber itself. And thus, we get to do stuff like this. We get to actually move around. So, okay, we know that actin and myosin pull on each other to lead to muscle contraction, but how does this all get initiated? Well, we have to start from the nervous system. One motor neuron, or a neuron from the nervous system, a nerve cell, connects to a couple of muscle fibers, all right? And that one motor neuron is the only thing that tells those muscle fibers to actually contract, right? And that's called a motor unit. So the motor neuron and all the muscle fibers it tells to contract is called a motor unit. Very, very important concept. We'll talk about it later in the nervous system section. There you go, motor unit, nerve, and actual muscle itself. So the way this process works, and we'll get much more into detail this week, we have already gotten into detail of this in class, the motor unit essentially, or the motor neuron, releases acetylcholine. This acetylcholine binds on receptors to the sartolemma, and we get a depolarization, or essentially we get an electric current flowing deep into the cell. That electric current essentially tells the sarcoplasmic reticulum to release calcium into that space of myofibrils where actin and myosin are. What the calcium does is the calcium removes troponin, all right? It removes troponin from actin, and that moves all the tropomyosin off of what are called actin active sites. And myosin heads really want to slide onto the active sites and pull them over. But they usually can't because there's no calcium to free up those active sites. The sarcoplasmic reticulum gets stimulated by the nervous system to release calcium. And then once that calcium is there, tropomyosin slides off, it frees up those actin active sites. The myosin molecules bond to the actin, and they pull and pull and pull. So as long as the sarcoplasmic reticulum is releasing calcium and leaving it in the cell itself, we have muscle contraction. Now, of course, myosin doesn't just pull for free. The actual myosin head has an enzyme in it called myosin ATPase, which takes ATP and uses that energy to re-release and pull again. So we need essentially two basic things for muscle contraction. We need calcium to free up the actin active sites, and we need ATP to actually power that contraction. So the calcium flowing in takes the tropomyosin off the actin, allows that actin to expose its binding sites, myosin clips right in, and through ATP it clips and clips and clips and clips, so it pulls, lets go, and then you get the Z disc shortening by actin being pulled into the cell. Pretty straightforward. So we have some visual representations of this here. And of course, much more in detail in this discussion as well. This is all in your notes, of course. On to some more details. All right. Muscle fibers essentially all work in a very similar way. You get calcium release, the actin and myosin. The myosin pulls on the actin, the Z disc shortens, that causes ATP. Contraction occurs in a very similar way. However, there are two essential different types of muscle fibers. There are fast twitch muscle fibers and slow twitch muscle fibers. They work in a very similar way. The difference is, in a couple of details, mostly about how fast that contraction actually occurs. Slow twitch muscle fibers, which is our second one here, They're usually very small muscle fibers. They don't have a ton of myofibrils packed in there. They're just pretty small muscles. And the actual speed of the myosin contraction is pretty low. However, these myofibrils, or these muscle fibers, the slow-twitch fibers, have lots of mitochondria in them and a lot of other adaptations to have a lot of endurance. So they can't produce a ton of force. They don't produce a lot of explosive force. They don't generate a lot of power, but they can continue to perform the same slow and smooth and steady contraction for a very long time. They have high endurance. On the other hand, fast-twitch fibers are usually big. When you train them, they have the potential to grow even bigger. Each myosin ATPase contracts really rapidly and with a ton of force. So fast-twitch fibers are really good for weightlifting and American football, for tackling someone or sprinting really fast or jumping really high. They produce high forces very quickly, and they can produce a lot of force, but their endurance really stinks. They don't have a lot of mitochondria, and they can only contract powerfully for a couple of seconds, and then they get really, really tired. Slow-twitch fibers, not very big, not very mean, not a lot of power or strength, but they can contract over and over and over, so they come really handy for endurance sport, for example. And you have all the details here, essentially. All right. And here we can see, this is kind of a diagram. Essentially, you activate a muscle fiber with a probe so that we know the nervous system is not involved. And if it's a fast-twitch fiber, you can see that it rises to its peak tension very quickly. Boom, it contracts right away. A slow-twitch fiber takes its sweet time to get contracted, and that's okay because it's better built for endurance and not for explosive function. All right. And it's important to note, and we'll get into, we've gotten into this in class in a bit more detail, fast-twitch fibers mostly produce more force because they're bigger. But even per cross-sectional area, fast-twitch fibers are a little bit stronger than slow-twitch fibers. They're not just more powerful, not just faster, they actually produce more force. All right, so muscle fiber recruitment, or motor unit recruitment. If we want to lift a weight that's incrementally heavier, let's say we're pushing against a machine, and that machine is pushing back as hard as we push into it. You start pushing something, the first motor units that turn on are your small slow-twitch motor units. These are motor units that control a couple of really small slow-twitch muscles. They don't produce a whole lot of force at all. They're the first ones to turn on every single time. That's called the size principle. Once they turn on, they essentially go all or nothing. A motor unit is either on or not. There's no 50-50. So those motor units turn on, and let's say they're not producing enough force for us. The machine isn't moving. So then we turn on bigger and bigger and bigger motor units that have more and more fast-twitch muscle fibers. And there's a pretty good diagram right here. So if we want to use maybe 10% of our total ability to produce force, at first we're only turning on slow-twitch motor units. Then we turn on more fast-twitch motor units, and then finally the FTB fibers, the super fast-twitch giant fibers, those are the motor units we turn on for real emergency situations. So if you're jogging, you're only probably producing 30% of your maximal force or so. Most of your motor units that are fast-twitch, they're not even on. The slow-twitch ones are the only ones turning on because they're the only ones we need. The body never uses motor units that it doesn't have to have. But if you're trying to squat as much weight as possible, you're going to be turning on the fastest twitch motor units and all the slow ones as well. And remember, it's motor unit by motor unit. There's no such thing as a 50% motor unit activation. It's either on or off. All right, and to finish up the lecture, muscle fibers can be classified into a couple general types depending on the movement. So agonists are muscles that are responsible primarily for the movement itself. So if we're doing a bicep curl, the primary agonist is the actual bicep muscle because it provides contraction and moves our limb to the desired location. Antagonists are muscles that oppose that action and sometimes slow it down so that it doesn't get out of hand. So your triceps, which are designed to extend the elbow, are the antagonists in a bicep curl. Sometimes they prevent you from curling so fast you hit your face. So if you're producing a lot of force, your triceps will turn on just a little to smooth things out, but your biceps produce most of the force itself. And synergists are muscles that are located in other areas that might help the movement a little bit. So the pectoralis muscle could be a synergist in a bicep curl, so that when you curl, you're not flaring your upper arm in anywhere. They essentially hold the upper arm in place. Your lats will hold the other part of the upper arm, and you get a smooth contraction with this upper arm steady and the lower arm actually moving. So synergists help the movement out, but they don't provide a ton of force for the primary motion itself. Boom. You can take a look at that in your spare time. And generally speaking, there are 3 distinct types of muscle action. When a muscle is contracting and is shortening, it's called a concentric action. So we'll do this with the bicep. I'm contracting the bicep, and the bicep itself is shortening. That is concentric action. Isometric action is when we're still contracting that muscle, but it's not going anywhere. So if I hold my hand down and I still contract my bicep, there's no movement. The muscle's not shortening, but there is muscular contraction. That's called isometric. Iso for stable or no change, metric for movement. So there's no movement, but there is contraction. It's isometric. And last but not least, we have eccentric contraction. That's when, if you grab a dumbbell that's too heavy, the bicep is contracting, it's producing force, but it's lengthening. So you're still contracting, but the bicep is actually lengthening. That's called an eccentric contraction, so force generated while lengthening. When you're walking down the stairs, your quads contract eccentrically. They contract, but they still allow you to expand the quad so that you actually successfully walk down the stairs, but you're not falling down the stairs. And there's a good diagram for you to look at in your spare time. All right. So we'll talk about this in quite a bit more detail, but there are a couple of very important factors that modulate force generation. How much force are you producing at any time, or how strong are you is another question. First of all, the number of motor units activated. If you're only activating 50% of your available motor units, you're not going to be producing much force. So the number one ticket to producing a lot of force is to activate all the motor units you have available. Number 2, the type of motor units activated. If you activate the big fast twitch motor units, you're going to produce a lot more force. Muscle size, probably the biggest determinant of force. How do you check in the street if you're walking around and someone asks you, hey, is that person strong or not? What's the first thing you look at? Well, are they muscular? If they're muscular, they're probably strong, because muscle cross-sectional area literally has the actin and myosin to produce force. So fast twitch, slow twitch fiber, that's all secondary. If you have a big muscle, it's going to produce a lot of force. Initial muscle length, if you start a bicep curl out here versus here, you're going to produce a lot more force if the muscle length is optimal versus very stretched. And of course, joint angle relates as well. If you're at a very poor joint angle, it's hard to produce a lot of force. But if you're at a more optimal joint angle, you produce lots of force. Weightlifters and powerlifters and people who compete in strength sports know this, so they always get into the right position for their lifts, which helps them maximize the force production they can do. Speed of muscle action gets a bit complicated. We'll get into that in class. That's it for Chapter 1, or Presentation 1. See you next time for Presentation 2.